All right, in this video, we're going to do an introduction to immuno-oncology. And the idea here is we're trying to leverage the immune system in some way. There are a handful of different methods to try to treat cancer. And traditionally, cancer has been treated by chemotherapy drugs, which are small molecule inhibitors that block enzymes or signaling proteins somewhere in the cell division pathway. The problem is cell division happens in a lot of cells other than just cancer cells. And the hope of immuno-oncology as a field is that we can be more specific, right? So the idea is usually we're going to use an antibody or maybe a modified T-cell receptor and something like CAR-T, which I'll cover in a different video. And we hope that the antibody is very specific to a target that gives the tumor, the cancer cells, the abilities that it has. All right, so in all of this, we can leverage natural killer cells, we can leverage T cells, we can use all sorts of different structures, right? We can activate macrophages via antibodies, but all of this depends on having an antigen that is in some way specific to the cell that we're targeting. And so these surface molecules that are being shown, right, on the tumor cell, they have to be cancer specific in some way. And there are two major options there. One is these could be normal human proteins that are just overexpressed on the cancer cell, which makes them selective based on quantity, right? We're just going to have more impact on the cancer cell. Or the perfect solution is if one of these surface proteins on the cancer cell is mutated in some way and is slightly different than the normal, like healthy person version of the protein, then we have a novel antigen. And by novel antigen, I mean something specific that an antibody will bind that's not found on other cells, right? The more we can get that, the better specificity there's going to be, and the less severe our side effects are going to be, because the antibody won't recognize other healthy cells outside of the tumor as much. All right, before I get into the details of the example that I'm going to use, I thought this was an interesting thing to point out. So this paper is going through the concept of looking at single cells in immuno-oncology therapy. And by single cell, we mean a single cell within the tumor. The problem here is tumors are heterogeneous. That means that if you have a tumor that's made up of 10 million cells, they are not 10 million clones of each other. They're not all identical because mutations pop up over time. And so a tumor may have started with, let's say, 4 different mutations that allowed it to start growing out of control. But it may have acquired a 5th, 6th, 7th, 8th mutation at some point after the initial tumor started. And so those mutations won't be found in every single cell now. They're only going to be in a subset. Okay. So the idea here is if we have a tumor, right, and in this case, the tumor is being represented as a group of different cells, and each colored cell is showing you that they have slightly different mutations, slightly different problems that are giving them their cancer-like properties. And if you can do a suspension where you isolate one individual cell at a time and then extract either the DNA or RNA from that cell and amplifying it, right, we can then sequence the genetic material from that single cell to know what mutations it has. Right now, this is critical because these treatments are going to have to be specialized. There's no reason that an immuno-oncology therapy for one particular type of cancer would work for any other type of cancer if they didn't share that same mutation and the tumor wasn't using the same mechanism to grow. And so the idea here is we want to know not which cancers we can use the drug for. Even more than that, we want to know which specific cells within the cancer we can use it for. All right, because the hope that one drug is going to kill every single cell in a tumor is unlikely. It's probably not going to happen. All right, but if we can use a combination of drugs, right? Say in this picture, the orange and green cells respond to drug X, and the blue cells respond to drug Y, and the purple cells respond to drug Z. If we can give those drugs either in combination or in sequence, now we have a chance of actually eliminating the tumor because we're addressing all the different mutations that have popped up along the way. Okay. All right, so this gives a better overview of things. and put some context into where immuno-oncology fits into this entire process. Right, so we have our complex tumor with many different types of tumor cells, many different types of immune cells, surrounding cells that maybe aren't part of the tumor but are present, and we need to know how are they best going to respond. Right, so we might use surgery to first eliminate as much of the tumor as possible. We might use traditional chemotherapies if the mutations tell us that they have a good target. Right, we might use radiotherapy, right, like gamma radiation, to try to kill off tumor cells, and all of these other things. Immunotherapy fits into that category of treatments that have the potential for an effect, but it's unlikely that it's ever going to be used completely independently on its own, because we already know that these other methods work to some degree. So why not combine them, right, and add immunotherapy to our existing repertoire? This is why when you see clinical trials, it is very unlikely that you're going to see an immunotherapy being tested on its own. It's always going to be in addition to some pre-existing treatment that we know is already effective, and we're just trying to see if we can make it now more effective. All right, so as an example of this principle, I'm going to use a category of immuno-oncology therapy called immune checkpoint inhibitors. And the specific immune checkpoint inhibitor that I'm going to focus on is called PD1. PD1 is the receptor, PDL1 is its ligand that binds to the receptor. And the general concept for these drugs is PD1 acts as an immune suppressor. Effectively, it's a way for human cells to tell the immune system that everything's good and they can be left alone by T cells that otherwise might kill them. Right, so this would be our CD8 positive T cell. That's you might hear it called a killer T cell or a cytotoxic T cell. The idea is the T cell is scanning for mutated or damaged or foreign, like virus-infected cells throughout the body, and if it finds them, right, it's going to try to kill them. All right, so this PD1 PDL1 combo, all right, PDL1 is the protein on the tumor cell, and PD1 is the receptor that's on the T cell. If the PD1 receptor comes in contact with the PDL1 protein, that basically is a sign that says, leave me alone, right? This is effectively how healthy human cells are supposed to make sure that they don't get unnecessarily killed by the immune system. All right, so if we zoom out and look at this in context, this version is how things are supposed to work, right? Our T cell is supposed to recognize that this tumor cell has an antigen there that doesn't belong. That's being shown as this tiny little red circle here. All right, so when the T cell receptor recognizes a foreign antigen on the tumor cell's MHC, right, the foreign antigen would just be a mutated protein from the cancer cell, the T cell is supposed to release these toxins, right? Perforin and granzyme B are just toxic substances that are then supposed to overwhelm and kill the tumor cell, and this happens all the time. Every human on the face of the earth, very frequently probably, has cells that start to look like tumors, and T cells, if everything's working properly, find them, use this mechanism of recognition, and kill them, right? If the cancer cell starts overexpressing PD1, right, like we are here, this isn't going to happen. And you can see that there's a selective advantage here. This, I should have said the tumor cell expresses PDL1 protein. The more PDL1 the tumor expresses, the better it is at telling T cells to leave it alone. And so you can imagine the cancer cells that happen to make a lot of PDL1 have an advantage. They survive. They're less likely to be eliminated by the immune system. That's why we see them showing up in tumor populations the way that they do. Okay, so the point of our therapy is going to be, we want to prevent this from happening, right? And the easiest way to do that is to just prevent the T cell from being able to recognize, right, these PDL1 ligands. All right, now in principle, this could work one of two ways, right? So untreated, this is the same setup that we saw in the previous slide. Tumor cell is making PDL1, T cell is making PD1, which is completely normal, right? This T cell is recognizing an antigen from the tumor cell, which normally would make the T cell attack the tumor cell and try to kill it. If PD-L1 is present in a high enough quantity, that's not going to happen because we're inhibiting that process. So we've got two major compounds that could work for drugs. We could use drugs that bind to and block PD-L1, right, block the ligand, or we could use PD1 blockers themselves, right, block the receptor. Either way can possibly work, or you could use them in combination. Right? And so in the end, you're going to basically prevent this tumor from signaling to the T cell that it's supposed to be left alone. And the hope then is that recognition of the antigen by the T cell receptor will lead to attack, tumor cells will die, right? And we at least make some contribution to getting rid of the cancer, in addition to other treatments that are being used alongside with it. All right, and this slide summarizes some of those options. So in this picture, this yellowish colored structure is PD-L1. So option one is we can block PD-L1, and these are examples of drugs that have taken that strategy. This guy in blue is PD1 on the T cell, right? We can also use PD1 blockers. Notice all of these drugs end in MAB. That is because they are all monoclonal antibodies. See the Y shape? Antibodies are perfect because we can use an animal like a mouse or a rabbit to generate antibodies that we know will stick to these targets, right? And just to float it out there, it's not the main focus of this video. There are other immune checkpoint inhibitors like the CTLA4 molecule, and antibodies to block that have also been developed. Okay, so the hope in all of this is remove the immune checkpoint ability from the tumor, sort of set the T cell loose, and allow it to, sort of take the brakes off of its suppression, go after the cancer cell, and hopefully we shrink the size of the tumor. All right, and just for a quick follow-up from that, there are many clinical trials that have used this strategy, but as an example, this cemiplimab, again, MAB tells you monoclonal antibody. This is a PD1 blocking antibody, exactly like we just showed. And we're going to test that to see if it adds any improvement to traditional chemotherapy. So for these patients, you see traditional chemo gave this survival curve, right? Probability of survival meaning this was 100% of the population being alive at month 0. And then, unfortunately, because these people have pretty severe types of cancer, the survival rate is going down. And throughout the course of the study, adding our antibody in addition to traditional chemo increased survival rates at almost all points. Now, I realize that the survival curve still isn't great. We would obviously love to see something perfect where everyone survives. But the harsh truth of cancer pharmacology is it's a slow grind. Improving these therapies is very, very tedious. And anytime you can gain an improvement of a few more months of survival, it's considered a win. Okay. All right. Hope you enjoyed the video. Keep following this playlist because there'll be more both immuno and oncology related videos to come. And subscribe to the channel so you get anything new that's posted.